Glycolysis is a fundamental metabolic pathway that breaks down glucose into pyruvate, generating energy. It occurs in the cytoplasm and consists of 10 enzyme-catalyzed steps, divided into two phases, the energy investment phase and the energy payoff phase. 1. Energy investment phase, steps 1 to 5. 1.1. Phosphorylation of glucose. Hexokinase or glucokinase in the liver phosphorylates glucose to glucose 6-phosphate, consuming 1 ATP. This traps glucose within the cell and initiates the pathway. 1.2. Isomerization of glucose 6-phosphate. Phosphoglucose isomerase converts glucose 6-phosphate to fructose 6-phosphate, an isomer. 1.3. Second phosphorylation. PHOSPHOFRUCTOKINASE1 PFK1 phosphorylates fructose 6-phosphate to fructose -1, 6-bisphosphate, consuming another ATP. This step is a key regulatory point. 1.4. Cleavage of fructose -1, 6-bisphosphate. Aldolase cleaves fructose -1, 6-bisphosphate into 2,3-carbon molecules, glyceraldehyde 3-phosphate G3P, and dihydroxyacetone phosphate DHAP. 1.5. Isomerization of DHAP. Triose phosphate isomerase interconverts DHAP and G3P, ensuring both molecules proceed through the pathway. 2. Energy payoff phase, steps 6 to 10. 2.1. Oxidation and phosphorylation. Glyceraldehyde 3-phosphate dehydrogenase oxidizes G3P and adds a phosphate group, forming 1,3-bisphosphoglycerate. NAD+ is reduced to NADH. 2.2. ATP generation 1. Phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP, generating ATP, substrate-level phosphorylation, and 3-phosphoglycerate. 2.3. Isomerization. Phosphoglycerate mutase relocates the phosphate group from the third carbon to the second carbon of 3-phosphoglycerate, forming 2-phosphoglycerate. 2.4. Dehydration. Enolase removes a water molecule from 2-phosphoglycerate, forming phosphoenol pyruvate (PEP), a high-energy molecule. 2.5. ATP generation 2. Pyruvate kinase transfers the phosphate group from PEP to ADP, generating ATP, substrate-level phosphorylation, and pyruvate. 3. Summary and regulation. Glycolysis yields 2 ATP net, 2 NADH, and 2 pyruvate molecules per glucose molecule. Several enzymes, especially PFK1, are tightly regulated by factors like ATP, ADP, AMP, and citrate levels. This ensures that glycolysis proceeds at a rate appropriate to the cell's energy demands. The pyruvate produced further enters either aerobic respiration, Krebs cycle and oxidative phosphorylation, or anaerobic fermentation pathways, depending on oxygen availability.